Now, I think something we need to discuss here is the fact that automation, it may not be possible or even recommended for, all steps in the manufacturing process. Are what I want to talk to both of you about is, are there barriers to automation? Um, should everything be automated? And I'd love to hear your thoughts on that, Craig. Can we start with you?
Sure. Yeah. Again, I think if you just go back to the principles of automation, you never automate a bad process. So if you don't actually understand the processing step, and honestly, many of our programs are preclinical or phase one, you probably don't have a perfect understanding of the process and the process variables that actually matter yet. And you're still testing whether you have the patient outcome that you want to have. So those steps I shy away from, and I focus the automation on the media handling or other things to make it as robust and scalable as possible. I think the other step is if there's really low volume steps and you have a tight window of process design space, I shy away from automating that until the technology can handle really tight tolerances.
Okay. Narinder, what are your thoughts on that? Should everything be automated? And if not, why not?
I think Craig said it well. We don't automate a bad process for sure. But even for a good process, the goal is not the automation itself. The goal is to create a simple, scalable process that can be, that can deliver large amount of doses in robustly, right? So, in fact, in some cases, like Craig mentioned, automation can complicate operations. For allogeneic, you can possibly do automated visual inspection, bring more consistency, for example. But for an autologous product, you have a few bags per patient. Putting automation around visual inspection, which some people have tried, would actually make the process much more complex. And because the cell product is somewhat different, you're just talking about a few bags, easy to inspect manually, and just go on. So in this case, it's a closed process, there is no risk. In other places where the process is not closed and it's open, automation may be more value even for a patient-specific batch manufacturing.
Okay. I wanted to go back at the top of the call. I believe we talked a little bit about, we touched on the source material. When it comes to collecting source material, how does an automated process improve safety and efficiency overall, specifically? Craig, talk us through that if you could.
I think this is where the two type of processes diverge greatly, right? For allogeneic, when I talk about starting or source material, I go back to the classical cell banking steps. I'm not too worried about collecting the donor material or the reprogramming, but I go to how do we select a clone and how do we expand it out? And you can use a lot of the benefits of monoclonal antibody technology of creating cell banks. When you go to the autologous world, Narinder hits it as well. You have to figure out, are you doing fresh collection? Are you able to cryopreserve that selection before you ship it off, which has some advantages as well, but you may have some cell degradation. So each of those areas you have to balance what's necessary for your typical process. And then by keeping it as simple as possible, and if you're putting the automation at a donor collection site, then that obviously, if you keep it simple, you'll have less variability, and the variability will only come from the patient, not from the process of freezing it down, etc.
Okay. Narinder, what are your thoughts on this, from collecting the source material perspective, in improving overall safety and efficacy? What do you think?
Yeah, I mean, I think here we're talking about live cell material, so you cannot totally sterilize it, and definitely not with heat or radiation, but also not filter it. Right? The cells are too big to be still filtered. So in this case, and then you have cells being collected from patients who are sick and in different stages of diseases. So it's going to create a lot of variability. And then you're collecting from different hospital sites, different collection centers, which the industry has done a great job standardizing the collection process, but still there is that variability from collection center to collection center. So I think one really need to look at it a bit more holistically. I mentioned that apheresis collection and cryopreservation, if we can create a validated, automated solution that can be implemented robustly at these collection centers, that would be a game changer. Then once we get the product in a manufacturing facility, there's definitely more control. But in many cases, we have seen more open steps, and our goal is relentlessly working on closing down those open steps. And in case of certain reagents, we have, for example, vector, we have seen vector coming in vials, and then you have to now transfer the vector to the cell process. So how do you transfer that vector in a closed manner? It's challenging, and finding ways to working with those suppliers to have more closed systems implemented right from the get-go. That's what we are focused on.